My name is Linnea Fletcher, and I am the Executive Director for InnovATEBIO, and I'm very excited to be able to help host these in conjunction with Bio-Rad. And from this point, I'm going to let Bio-Rad do the rest of the introductions because they will be doing the presentations for advanced PCR.
So thanks, Linnea. I'm Bryony Rugg. I'm the director of the Bio-Rad Explorer program. And I'd like to introduce our speakers today, who are Dr. Liz Dreskin, who is an R&D manager at Bio-Rad, and Dr. Josh Steele, who's a microbiologist at the Southern California Coastal Water Research Project, which is shortened to SQUIRP, which I'm sure you'll hear him say later. I will do Josh's bio once in his part, which is coming in the second part of the talk. Right now, I'll tell you a little bit about Liz. So Dr. Dreskin oversees the applications team for the Life Science Group at Bio-Rad. Her team works with the marketing group to develop the data and literature and application notes that help users use the Bio-Rad tools, both the instrumentation and the reagents and the software for all the different products in the life science range, including electrophoresis and Western blotting, PCR, quantitative PCR, Droplet Digital Pcr, next-gen sequencing, electroporation, flow cytometry, and protein purification. So we're very glad to have Liz speak to us today about PCR in general, and then to give us a little bit of a deep dive in Droplet Digital PCR. Thank you, Liz.
Thanks, Bryony. I'm gonna share my screen and then get this started. Okay, so I'm just going to give a brief overview of PCR, how it works, how PCR, quantitative PCR, and Droplet Digital PCR are all related, and then a little bit about conventional PCR, Droplet Digital PCR, and then next time, real-time PCR. So it's hard to believe that PCR was invented less than 40 years ago, but it's had a huge impact on life as we know it. It was invented in 1983 by Kary Mullis, who was at Cetus Corporation. And 10 years later, he received a Nobel Prize in chemistry. This invention opened up lots of areas of exploration for scientists, and it's affected so much of our lives. So real-time PCR was invented in the early 90s, and then Droplet Digital PCR was invented around 2010 or 11. It's affected medicine and opened up lots of areas of research for us. So just a brief overview of PCR and the components that are required. Taq polymerase, I'm going to just call it Taq polymerase. There are many companies who have worked on different modifications of Taq polymerase. So I'm not really referring to the original one. This is just a generic Taq, but companies have designed their own Taq polymerase to have different features. Along with the Taq polymerase is a reaction buffer to allow the polymerase to work optimally. Then we have a DNA sample. We have forward and reverse primers that are complementary to our sample. Deoxynucleotides to allow for the extension and copying of the sequence, and then whatever reaction vessel people want to use, small tubes, 96 well plates, 384 well plates. So PCR is a multi-step process. There are basically 3 steps within one cycle. The first being denaturation, in which the strands are separated, and then annealing which allows primers to bind to the strands, and then extension, which allows the polymerase to go and extend those to make copies. In a typical PCR process, about 40 cycles are done. One other thing I want to mention about PCR is that I'm referring to all of this meaning hot start. So you may have heard the term hot start, and basically the polymerase is being reversibly inhibited from working at room temperature by the addition of an antibody or aptamers or chemical. And this is because we don't want the polymerase to start working while our reaction is being set up, because we might get non-specific products and primer dimer issues. So hot start refers to an initial denaturation step at 95 degrees that occurs prior to the cycling. And that step could take 2 to 15 minutes, depending on the company's formulation of their tech polymerase. Briefly want to mention the power of PCR. So with every cycle, the number of copies of your DNA is doubling. So after 30 cycles, you have over 1 billion copies. So that's a huge amount of template that you could do different things with. And we can talk about that in a minute. What happens if you want to quantitate messenger RNA to look at protein expression? First, your RNA has to be converted into cDNA. So this is an example of a messenger RNA with a poly-A tail. We'll need to anneal primers like oligo-DT, random hexamers, or gene-specific primers. Add a reverse transcriptase, usually which has RNase H activity associated with it, and dNTPs. The RNase H is important because it destroys this original RNA copy as it's being reverse transcribed, so that we keep our ratio of our starting material. And then we end up with a first strand cDNA, which can be used as a template in our PCR reactions. So in this table, we've got a lot of information. I just want to point out some of the benefits and drawbacks to conventional PCR, digital PCR, and quantitative real-time PCR. So conventional PCR is relatively inexpensive, it's low tech, and you get a yes or no answer. Droplet Digital Pcr is quantitative, it's extremely sensitive, and it's tolerant to PCR inhibitors. Real-time PCR has a broad dynamic range, allows for higher throughput in that you can use the 96 or 384 well plates. And the drawbacks are that conventional PCR is semi-quantitative at best. Droplet Digital is a little bit more expensive, but also less expensive than other techniques like NGS that are out there. It has more steps and longer overall time to result. Quantitative real-time PCR requires a standard curve to be quantitative. So that takes up some of your real estate on your plate, so you can run fewer samples. It also requires some skilled labor for analysis, the same as digital PCR. All of these techniques are complementary in that you may need to use any one of these techniques to answer a specific question. So let's talk about conventional PCR. Some of the areas where conventional PCR is being used include forensics, food and agriculture, environmental microbiology, phylogenetics, population studies. Some examples of equipment that you might find in a lab doing conventional PCR are cyclers from Bio-Rad, Thermo Fisher, QIAGEN, and Eppendorf. And PCR is an endpoint experiment. We're looking for a result after the cycling is complete, and almost always by running it on a gel. Gel electrophoresis, as you know, is a technique to separate DNA fragments based on size. This is an example of an agarose gel. You can use this to see if you've amplified the correct product. And how do you know if you've amplified the correct sequence? You don't always know, but it'd be great to include controls. And so a negative control is always important, and positive controls, if you have them, are very helpful in figuring out what happened when you don't see the result you were expecting. I'm showing an example of multiplex PCR here. In the upper cartoon, we have 4 individual assays, and after the amplification, they're run on an agarose gel, and you can see the 4 different results. If the primers have been designed carefully and the amplicons are of different sizes, you can actually combine these into a multiplex experiment in which all 4 of these assays can be used in one tube to look at the sample. This saves sample time, sample size, and also space on your gel, so you can get higher throughput this way. Some other types of PCR that you may have heard of include nested PCR, asymmetric, inverse, gradient PCR. Two-step PCR is actually quite frequently used, and it's the replacement of steps 2 and 3 in that 3-step PCR that I showed you earlier with 1 step. So it's 1 step for anneal and extension, and the extension basically happens at the annealing temperature. Loop-mediated isothermal amplification, also called LAMP, I'll mention in a minute, and then digital PCR limiting dilution, I'll also mention that. So this is a diagram of LAMP. The key point is isothermal. It happens at 1 temperature. So a thermal cycler isn't really needed to do this experiment. But we just need to hold this at 1 temperature. I've taken this schematic from NEB, which shows that LAMP requires 4 to 6 primers. These primers have incorporated a complementary section so that they will end up forming a dumbbell-like structure once they've been incorporated into the product, and also a DNA polymerase, which is strand displacing. So once there's a primer bound to a template, the polymerase can go through and separate the strands as it's synthesizing a new strand. There are several methods to detect the LAMP reaction, and this is just an example of one of the products, but there are so many different products that can be formed from this type of reaction. This is one example of a colorimetric readout. You could also have fluorescent readouts and other ways of detecting the products. But the beauty of LAMP is that it's fast, and you can incorporate dyes so that you have a visual result. You have a yes or a no answer, and it happens very quickly. This is often used in rapid pathogen detection, requires very little equipment, so it could be used in the field or in developing countries. It's currently being used for SARS-CoV-2 detection. It's been used for Zika virus and influenza detection as well. And now I want to talk very briefly about digital PCR, limiting dilution. This is an idea that Poisson distribution statistics can be used to quantitate the starting material by knowing how many samples we have that are positive and calculating the statistics on them. So in this example, I've got 100 copies of gene B. Let's say it's in 5 microliters. If I dilute this tenfold, I'll have 10 copies in 5 microliters. And then I dilute another tenfold to have 1 copy in 5 microliters. Now, if I take 5 microliters from this sample and assay it, I won't always detect 1 copy. In fact, the probability that I'll detect 1 copy is about 37%. So if I were to fill a 96 well plate with reactions from this dilution, I would only expect to see positive or 1 copy in 37% of the sample of the wells. There are also going to be some wells that have more than 1 copy, but we're not interested in that. This is an endpoint detection. It's a yes or no. So now we can move to Droplet Digital PCR. So we talked about looking at that dilution where we could assay the 5 microliters and maybe we would detect something and maybe we wouldn't. If we want to consider digital PCR as a method of further partitioning the sample, so instead of pipetting 96 different wells, we could actually take this reaction and create thousands of little droplets. And let's say 20,000 droplets. It becomes easier to find the needle in the haystack when you've got 20,000 droplets. It increases your precision because you're measuring thousands of times. And then it also increases the sensitivity by increasing your local abundance. Now, Bio-Rad uses droplets to perform this partitioning. Other companies do this by micro arrays or nano arrays. But the idea is partitioning the sample. Some of the equipment that you might see in labs doing droplet digital PCR or digital PCR are from Bio-Rad, QIAGEN, Thermo Fisher, and Steele technologies. This is an example of the Bio-Rad Droplet Digital PCR workflow. So we start by preparing a reaction. This is a PCR reaction with one difference. We're adding fluorescent detection reagent. So that could either be another primer that's been labeled with a fluorescent label, or it could be a DNA binding dye. Once we have our reactions assembled, we can put a plate, in this case, into our auto droplet generator, which has a specially formulated oil that is combined with our samples to create about 20,000 droplets for each one of these wells. Once the droplets are generated, we go and seal the plate, and then we put it into a conventional cycler and amplify the targets. That then gets moved into the droplet reader, which can assess each one of these droplets, similar to flow cytometry, for a yes or no answer. It's positive or it's negative. And then we can analyze this by specialized software. We also have another instrument that combines most of these features into one, so it's more hands-off. So how is the data analyzed? Positive droplets have higher fluorescence than the negative ones. So our negatives are counted as zeros and the positives are counted as 1. This is digital signal. And this gets taken into the software, which calculates statistics on each sample. We can have also we can look for 2 targets or more than 1 target at a time. And this is an example of a duplex reaction where we're looking at 2 targets, each in a different channel or color. And in this case, you can see that each sample has both targets, but the channel 2, the green dots, are decreasing in these samples, while the template for channel 1 is pretty much held constant. Another way of looking at these data for a multicolor experiment would be in a plot like this, where you plot the amplitude of channel 1 versus the amplitude of channel 2. The key points are in the lower left quadrant, the negatives appear, and upper left quadrant is where the positives for channel 1 are. Positives for channel 2 are in the lower right quadrant, and then the upper right quadrant will have double positives. And the software will calculate statistics for each of those quadrants. I'd like to switch and talk briefly about liquid biopsies, what they're useful for, and why they're done. So liquid biopsy is a blood draw. And the reason it's so useful is because there are a lot of solid tumor cancers that are inaccessible or difficult to access for biopsies. Maybe you want to study somebody's the progression of somebody's cancer or disease, but the cancer is difficult to access. So we can take advantage of the fact that all blood samples from people contain cell-free DNA and cell-free RNA. In addition, cancer patients have circulating tumor cells in their blood samples and circulating tumor DNA. So in the case of a liquid biopsy of a cancer patient, this circulating tumor DNA is the needle in our haystack of regular cell-free DNA and cell-free RNA. This is an example of a liquid biopsy that's been used to detect mutations in solid tumors. It was determined that EGFR, a mutation in EGFR correlates with resistance to EGFR tyrosine kinase inhibitor treatment. So the need was to develop a 2-color assay looking in 2 channels, one for wild type DNA and another channel to look at the mutation. So this assay could be used to look at liquid biopsies from patients, and the clinician can look at the percentage of mutation found relative to the wild type and understand whether the treatment is still effective or if it needs to be changed. One other different use of digital PCR is in biosimilar purification. My group did a purification of the biosimilar rituximab. Biosimilars are similar to the biologics, and biologics are often antibody drugs that are used to treat cancers and different diseases. A biosimilar can be made when that drug comes off patent. So it's quite popular in biopharma and biotech. And these biosimilars are produced in different cells. In this case, ours was produced in a human cell line. They're often also produced in CHO or E. coli cells. But it's important that the host cell DNA be quantitated, because the FDA has rules about how much could be present in the purified product that you would treat a patient with. So we've assessed the loss of host cell DNA during our purification process so that our final we've lost all of our host cell DNA. We can't detect it anymore. Just to show some key advantages of digital PCR, absolute quantification without standard curves. It's very, very sensitive. You can detect rare targets in complex backgrounds like the liquid biopsy, and is tolerant to PCR inhibitors. This is just a sampling of what digital PCR is being used for. Rare sequence detection, environmental studies like you'll be hearing about next, mutation detection, cancer monitoring, organ transplant, prenatal diagnosis. And with that, I will take any questions. Thank you.
Hey Liz, thank you. Linnea did ask the question about how the droplets are actually generated. Can you go into a few details about that?
Sure, I can try. So I can think about our original droplet generator, which actually had a vibration to it and then forced these droplets through a small opening to kind of force the sample and the oil through a small opening to create droplets. That was actually the video that I couldn't find and we didn't include. Maybe I can find a video and add that to the link. But I would say that a lot of mechanical engineers were involved, and also the chemists who developed the oil to make these little tiny droplets that would encapsulate mini PCR reactions.
Thank you. That's really impressive, Dr. Druskin. Because I was just thinking about the methodology to just develop the droplets such that they would have the ability to have those products or substrates within each droplet to be used for this.
Yeah, it's really creative, and it's a whole group at our digital biology center that works on this. And I can't even fathom how they got to this point, actually, but it's pretty amazing.
Well, I just want to say for myself, your presentation has been absolutely amazing, and I really appreciate your willingness to take the time to do this for our audience.
Oh, you're welcome.
Any other questions? I don't think people always realize how much Bio-Rad does in terms of clinical applications. So I think this was also really good for that, because I think they forget about all the different products that Bio-Rad makes and their applications. It's not just education.
True.
Yeah.
A lot of products.
Do you mind me asking how you got involved with Bio-Rad? I know this is kind of a side topic, but I know that our audience might be interested in that, especially since they can relay it to students.
Sure. I actually probably have a slightly different path than most R&D scientists. I decided to leave the lab after my postdoc, and I joined Bio-Rad as a tech support specialist. And I just love tech support, interacting with our customers, helping teach them how to use different equipment or technologies, learning the new technologies as they were developed also, was really great. And then I eventually felt I needed to get back to the lab. So that was my path.
Well, thank you very much.
You're welcome.
Well, I turn this back to Bio-Rad.
I'll stop sharing.
Thanks, Linnea, and thank you so much, Liz, for this. I'm very much looking forward to learning more about quantitative real-time PCR in the next couple of weeks. I would now like to introduce Joshua Steele. So Dr. Steele specializes in environmental microbiology and microbial ecology. His research involves the development and application of molecular techniques to detect and track pathogens and natural microorganisms in coastal ocean and watersheds. Through the application of next-generation sequencing and metagenomics, he works to connect the environmental microbiome and antibiotic resistome to water quality, ecosystem health, and health risk, and uses bioinformatics to measure natural populations and community-wide changes in genes or gene expression in response to environmental changes. So he'll give us some really nice applications on how he uses PCR and digital droplet PCR in the field. So welcome, Josh, and thank you very much for your presentation ahead of time.
Well, thank you for that introduction. Let me see, I'm going to share my slides. Okay, can everybody see? All right. So yes, my name is Joshua Steele. I work for the Southern California Coastal Water Research Project, which is a bit of a mouthful, but it does have the lovely acronym of SQUIRP, which is easy to remember when you hear it, but not exactly easy to remember what it stands for. I'll start by doing a bit of a background on what SQUIRP is and what we do. We are a research institute, basically, for aquatic sciences that tries to improve the management of aquatic ecosystems in Southern California. And although we're focused in Southern California, we don't limit ourselves to Southern California. We try to do applied research in environmental coastal systems worldwide. We were founded as an intergovernmental public agency in 1969. So we've been going for more than 50 years. And we try to develop tools and technologies and strategies to help the water quality management community protect and enhance ecological health in the coastal zone. So that includes the ocean, the watersheds. And we are intergovernmental in that we are a collection of local government agencies, sewage treatment plants and wastewater treatment plants and sanitation agencies, as well as stormwater agencies and regulatory agencies. So all combined make up our commission, our board, basically. We cover a wide range of research, but one of the one that I'm most intimately involved in is microbial water quality research. And we primarily focus on identifying and locating sources of human fecal contamination in watersheds or in beaches. We are also try to measure pathogens directly in water in particular. And we aren't just restricted to beaches or stormwater. We also look in wastewater, which is what I'm going to be mostly talking about today. And we've been starting to branch out into other things like shellfish and anything that can basically affect the coastal zone. And ultimately, we translate this to human health risk and try to improve water quality to reduce human health risk. For what it's worth, we were very keen and early adopters of quantitative PCR and digital PCR, and we apply it in every one of these aspects. And I'm going to talk to you today about some of our work that we've been doing recently with wastewater-based surveillance of Sars coronavirus 2, and using digital PCR and some of the different things we've done with that. So you can feel free to interrupt me if you'd like with questions, or you can, if you want to wait till the end and ask questions, I'm always happy to hear. But I'm going to sort of give you a whirlwind tour of what we've been doing with the coronavirus wastewater-based surveillance. So, and you can hear a cat in the background since I'm working from home today. So a little bit of background on wastewater-based surveillance. The COVID-19 pandemic has put this technique, this method of wastewater-based surveillance back in the forefront of public health. this was once a very important part of public health surveillance, particularly when the poliovirus or enteroviruses were going along. But as the poliovirus has been getting closer and closer to eradication, it has sort of faded away until recently. In California, the wastewater community has been very active since the beginning of the pandemic. We have more than 40 facilities that have been monitoring, and they have at least 6 months of data. Some have been monitoring for over a year and a half now. And then nationwide, this has also been a big push, and we've been participating in national studies, as have some of the agencies in Southern California that we've been working with, and the national research community that's sprung up to try and monitor this. And we, along with the California Department of Public Health and the Water Quality Board, have been trying to sort of provide guidance on data use and interpretation, as well as more effective ways to monitor. So that's been our main focus. Oops, there we go. The reason we focused on methods and data use is that initially everyone jumped in trying to get rapid answers, and basically everybody used their favorite method and did it as fast as they could, because clinical results were lagging, and it was really important to try and find out what was going on. As we've moved on into new phases in the pandemic, the questions have evolved. And the public health community in particular has said, okay, well, we think we have a handle on some of this clinical data when we have high prevalence, but when we have low prevalence, or maybe when we're looking at how variants are moving through the community, that's where we think wastewater-based epidemiology, wastewater-based surveillance is going to be really important. And so we've tried to focus, in addition to monitoring, on method variability and sensitivity. And I'll sort of give you some examples of what I mean and what we've been doing and how that relates to digital PCR and how we use it for that. Don't worry, I'm not going to go through every single one of these potential sources of variability, but this is just sort of in flowchart form, all of the variability that can happen when you're trying to sample. Well, in this case, it's for wastewater, but it really works in any situation. You have everything from your sample type and strategy and frequency to how you process it, whether you inactivate the pathogens, how you capture them, how you purify the nucleic acids, what genes you select, and then how you actually quantify it in the end. Again, don't worry, we're not going through these step by step. I don't want to lose all of you this quickly into the talk. But we started there where we were comparing all these different methods, and we've published on that in a couple different places. And we decided to try and figure out how we can translate that to more effectively using the data. So in particular, we wanted to look at method variability, and I'll show you what I mean by that, sensitivity, and then apply that to monitoring both just the overall quantity of viruses in the wastewater, as well as the variants in the wastewater. And that's sort of the variant is where we're going next. So one of the sources of variability was initially the gene targets. There were multiple primer sets available. Again, everyone sort of threw what they could at it. Labs had to make a choice and basically just picked one and ran with it. So this is a schematic of the genome for SARS coronavirus 2. It's somewhat small, but what you can see here is just like Elizabeth was talking about, you have your sort of sequences and probes when you're looking at quantitative PCR. That's what these are. You have the arrow going to the right is your forward primer, the arrow going to your left is your reverse primer, and that box in the middle is your probe. And so these are just a handful of the different assays that were designed to try and measure the coronavirus. So what we wanted to do is take a look at, we found the N stands for nucleocapsid, and that's the actual viral capsid, like the outside of the virus, that the genes for making that, which is really important. You can't make more viruses if you can't make a new capsid, so they should be a pretty conserved gene to target. And so we wanted to look, does it matter which of these different ones that we target? Does it give us a different answer? And so we went across multiple different treatment plants, and in a few I think this is a few hundred samples. They're just kind of all buried on top of each other. And what you can see is we had an incredibly tight correlation when we measured by digital PCR for N1 and N2 genes. So this was really good news. This made us feel good. We were looking at both of them at the time. And one thing that was curious, we have a slightly higher, if you notice, this isn't quite a one-to-one line. We had a slightly higher number of N2 and compared to N1. Other parts of the country, they noticed the opposite trend, a slightly higher number of N1 to N2. We don't know which one is the true measure. We've continued measuring both. So that's one place where the variability was fairly small. And I do believe that had to do with digital PCR for that matter, that we were able to get such a tight correlation between the two. All right, what about sensitivity? This becomes really important when the prevalence goes down. When the prevalence goes down, people also are less worried about it. And so perhaps the number of people going and getting tested with a clinical test also is reduced. So then the wastewater can be a way to monitor it when you have maybe spottier data from the community. We came up with some recommendations to improve the sensitivity. They're mostly not important to understand the details behind it, but basically how you inactivate the viruses can improve your sensitivity. If you're doing pasteurization or heat inactivation, that can reduce your measurement of the viruses. If you're trying to measure just a very small volume and directly extracting it or directly measuring it, that can reduce your sensitivity because you're just limiting yourself to how much of it you're measuring. Whether you're using digital or traditional qPCR, we found that digital PCR is more sensitive. And whether you're using sludge or influent, sludge tends to be more sensitive. It's again concentrating things down. So one of the places we took a look at this, we actually had an opportunity to split samples at one sewage treatment plant, and we had a small volume qPCR, a small volume qPCR where they concentrated it, a large volume where they were about probably 100 times more than the small volume ones, and then a large volume digital PCR that we did where we were about 20 times more than the small volume ones. And what you're looking at here are results, and you can see that the sort of medium volume, I called it large, but it's more of a medium volume digital PCR is the green line, tracks really, really well with the large volume qPCR. The other two had a lot of problems, and when they dropped to 0, that means that went below detection. So that means that their sensitivity was not, they were not sensitive enough to be able to detect it. And so this is a problem when you're starting to get down to lower parts of the curve where where you have less and less virus around. If you're not sensitive enough, you're not going to be able to follow it and get any information out of the wastewater. And if you zoom in to sort of that early part of the curve where it's low concentration, again, you can see the digital PCR and the large volume qPCR did a really good job and tracked each other pretty well. And some of these others, you had 1, 2, over 60% of the time they weren't able to measure it. And similarly, this other one, which wildly varied between not detecting and detecting very high, again, had a lot of trouble in that low concentration. So that matters. And that's something that we've now tried to apply to our monitoring and tried to recommend to other people to apply. So, the takeaways are that these larger volume and digital PCR do a good job at low concentrations. The smaller ones are really too insensitive to be useful, and our recommendations are looking all right. So I've gone over really briefly a few of the examples of how we compare the method variability and the sensitivity. And now I'm going to sort of show you how we've applied it to monitoring SARS coronavirus in wastewater. So what you're looking at here is a time series and the top plot is a average of that N1 and N2 gene. We took both of them and averaged them over time at a treatment plant in Southern California. starting way back in April of 2020 and going through February of this year. So you can see we had sort of the initial summer peak in 2020, and then it went back down to really low numbers. We saw a little spike in here that we picked up in the wastewater. And then as cases climbed up, we also were able to follow that. And we had a remarkably good correlation between the two of 0.84. We were really, really excited about that. And so it's been a really good way of monitoring it through the community of the overall prevalence of the virus. For what it's worth, I think I believe this one is 400 million gallons a day of sewage through this plant. Forgive the fuzziness of this one, but this is a different plant, and here these aren't average. These are the N1 and the N2 sort of plotted on top of each other, and you can see how well they correlate. But again, this is a different plant, but you can, and a little bit later, so this is starting in November, and you can see again we have that winter spike we saw in California, and then it went down during the spring and early summer, and then that secondary spike, again, we saw it come up in cases, and we were also able to track it, and it did a reasonable job there with a correlation of 0.76. So we were pretty happy with that. This just showed to us both the sensitivity of the digital PCR method we were using and the precision, and we were quite pleased with that as well. One other thing that we did, because there is some noise in the data, and as you might know or might imagine, sewage is not a constant mixture. It can change during times of the day, can change during times of the year. And so we're not interested necessarily in sewage that, we're not interested in tracking false patterns where maybe suddenly it's more dilute because there's more water in the sewage and less human fecal material. So we picked a viral human fecal marker, which is the pepper mild mottle virus. This is not a human pathogen. This is a pepper pathogen. But we find it everywhere we look in Southern California, and frankly, it's almost everywhere we look in the country. We all eat a lot of peppers and hot sauce, apparently. But it passes through us when we eat anything made from peppers. And so what you can see is once you scale it, you can actually somewhat improve the correlation at one of the plants and get basically a similar correlation, although again, not as much improvement there when you do it one-to-one. Without any smoothing, to try and see, well, how much SARS coronavirus per amount of fecal material is there. And so these are some of the ways we've been using these, and we can measure all of these in the same sample across it using the same methods, basically, the same reverse transcriptase digital PCR. So really quickly, I'm going to touch on variant analysis. Variants are tricky because wastewater, as you just saw there, is complex and noisy. And in addition to just its normal wastewater complexity, variants themselves, there should be multiple variants in the sewage most of the time. The virus is constantly mutating, constantly making new variants, and a single mutation may not be enough. You often have one mutation that will be meaningless. You'll have another mutation that narrows it down to like, well, okay, we have this lineage of variants like alpha, beta, and delta all share this one mutation. And then within that, you can say, okay, well now delta has this one, alpha has this one, and you can sort of start piecing it through place by place. And the way we learn about these is by sequencing in the first place, mostly from clinical samples, but we've also tried sequencing in wastewater to sort of see what the different things are. And then this interpretation has to be a comparison between the clinical data, so the public health agencies, the sewage treatment agencies, and researchers. It's a very complicated problem to solve, but we think we're making some initial progress at it. Again, here's this genome. They're really small numbers because they want you to actually look at their primers. But you can see on the primers too, there's 13,342, 18,778. All those numbers are locations on the genome. that they are targeting. And every single one of those locations could have a mutation. And so we recently published a paper with collaborators at UC Irvine. And this is just a handful of the mutations we saw. And this is a proportion of a mutation at any given position. So 241 is position 241 on that genome. So that one gene, 27,964, that's the position on the genome. And then the numbers on the x-axis are proportions to the wild type. So this mutation compared to what was in the original genome that they sequenced. And you can see that looking over time, you'll have rapid changes in these mutations. Sometimes it's completely mutated the whole time. Other times it starts out with no mutations. You see a big jump to everyone we measured was mutations, and then back down to the original wild type one. So this is basically just showing you how complicated it is, how many different mutations can be present in a mixed sample, and how difficult a problem it is to solve. However, building off of sequencing of clinical samples, where you don't have the mixture, you have basically one genome at a time, there's some hope. And the way we have to do it then is we identify this mutation or combination of mutations and attribute it to a variant. So for instance, the Delta variant, there's one that we have been testing that actually the primer and probe set spans both a variant at a specific location as well as a substitution variant. And it gets two variants in one primer set, and that enables us to call it Delta, or at least we think it does. And then we have to test it, make sure we're not fooling ourselves, and look in the sewage. Unfortunately, I don't have a ton of data to share with you about this, but what I can show you is the clinical data that we're building off of. And you can see here that, and this is in California, starting in last fall, the alpha variant started to come up and made 60% of the clinical samples. And then it started to fall off. It was being replaced, I'm sorry, the epsilon variant first, and then it was being replaced by alpha earlier in the spring. And then everything has been replaced by Delta. And while I unfortunately can't show you the data from the sewage treatment agencies from the sewage on that, what I can say is that we were able to track, we haven't gone back and looked at epsilon, but we were able to see the alpha, the rise and fall in alpha, and the replacement in Delta in the few sewage samples we have looked at. So where we're going next, we have our, we're continuing to look at variant research, and we're reaching out to our collaborators, all of those different groups we have to work with, and quantifying using this digital PCR, and working with the clinical agencies to see sort of what is coming next, like Mu or some of the others that they're concerned about, if anything can displace Delta or future variants of Delta, to try and track that in the future. And with that, my whirlwind tour is over, and I'm happy to answer questions.
Wonderful. Thank you so much. That was fascinating. I had a hard time following the check, so I was staring at all your slides, but we do have a bunch of questions. I might be a little out of order, maybe just to string the story in a way. Most of the questions surround the use of wastewater. So the question is, why just wastewater and not drinking water? And then questions about stability, about RNA in wastewater and any kind of PCR inhibitors that might be there.
Okay, those are great questions. So the first question of why wastewater and not drinking water, basically we're looking in this case at influent, so untreated wastewater. Treated wastewater, as well as treated drinking water for that matter, does a really good job of killing the virus. And so they were not particularly interested in that aspect of it as much. So they were much more interested in what was coming in as a way of tracking the virus in the population rather than being worried about it. To be perfectly honest, from what we've seen and from what other people who have tested it where they try to grow virus out of wastewater, just the soaps and detergents from your showers and your washing machines and your dishwashers is probably enough to kill the virus, even in influent. So it's not much active virus once it gets into that water. If it was poured in, if you had virus poured into drinking water post-treatment, I don't know what would happen. But treated water, very safe, not a threat. So it's really more a way to track what's in the community than worrying about what's coming in the water in this case. And then let's see, the next two questions were inhibitors and RNA stability. Yeah, the RNA stability is a really good question. I think the viruses themselves, as long as they're not broken open, they can be inactivated by those detergents just because their envelope gets disrupted, but that nucleocapsid I talked about stays whole. That will protect their RNA. So we actually use bead beating and mechanical lysis as well as chemical lysis to break them open and get to the RNA. But I've not seen anyone do a good test of free RNA versus viral capsid RNA in wastewater to see what the difference is. But I would expect it to get chewed up pretty quick. Mm-hmm. And inhibitors, yes, wastewater has lots of inhibitors. And that is one of the things we think is helping us with the sensitivity. In our with the digital PCR we use, some folks have been trying to spin out the solids and just look at the influent in the water itself. We don't. We look at the whole suspended solids and the water, and I think that digital PCR helps us just by the nature of well, I guess it's we think it works in two ways. We think one way is that the digital PCR by encapsulating the reaction in a very small volume, you can exclude some inhibitors that way. But honestly, I think part of its benefit is you're really counting positives and negatives. And so even if you're inhibited, if your positive is not this high, is not 5 times higher than your negative, but is only 2.5 times higher than your negative, you can still distinguish it and count it. And then and as a 1 versus a 0. And that digital PCR has a benefit that way. And not just in wastewater, when we've applied it in stormwater and other really difficult matrices and environments, we've found it to be a little bit more robust. I think we did do an experiment. My former colleague at SCORP, Yiping Cao, did an experiment where she spiked in inhibitors, and she was able to get it enough to where she did kill the digital PCR reaction, but it took quite a bit more than what killed a qPCR reaction.
Okay, another question. Could you please explain specifically what is larger volume concentration qPCR?
Yes. So I was using some shortcut categorization there. What they do is they take, I think, 200 to 500 milliliters, maybe larger, of wastewater, and they actually put in a large quantity of salt as a preservative, and then they put in a silica milk, and they do the extraction directly on that large volume in the silica, and then they take the DNA from that silica and then go into a qPCR reaction with that. So it's a very clever method they use, and it worked really, really well, as you can see. But I think as far as it's one that they've been able to scale up, I'm not sure it's one that would be easily scaled up for monitoring in the long term. It would be a tough one to automate.
Could I ask a quick follow-up, Yolanda, on that one before he leaves that? So have they gone and determined the sensitivity for large volume concentration qPCR with DNA standards so they know how sensitive it is?
So, I mean, they do spike, they do that 2 different ways. They do a sort of a spike in control with an RNA virus and with RNA itself. And then, yes, they do still run a standard curve. The sensitivity they've found is getting down to around 1,000 copies per liter, which is about the sensitivity we can get to pretty relatively easily. They could probably play around with volume and get higher. But as I really briefly mentioned, we are concentrating 20 milliliters on a filter and getting a similar sensitivity that they're getting with 200 to 500 milliliters.
Thank you.
Mm-hmm.
Okay, if anyone has a question that hasn't been addressed yet, feel free to add it. I think there was one more, and I believe you addressed it about, is the virus live in the wastewater, and have you done any plaque assays or the like to correlate the PCR data with other parameters? Is this a question having to do with, like, how do you know whether, okay, so we're seeing the virus in wastewater, does that mean that that's an active infection, or is that somebody basically at the tail end, like, pardon the pun, how do you know that has anything to do with active infections versus just residuals?
So the only way we've been able to address that is by comparing to case counts from the public health data.
Mm-hmm.
There are researchers. We do not have the biosafety level to cultivate this virus. There are very few labs that do. Those that have, a few of them had said they were going to look into wastewater. I have only heard anecdotally from some of them that nothing grew out of it. But they've honestly been a lot more worried about growing the virus out of clinical samples than worrying about the wastewater, since it doesn't seem to be a big source of infection. And the rates of people who work in wastewater treatment plants have not been any different than the general public. It does not seem to be spread via wastewater. And then the second part, yeah, we don't really know if the if people seem to shed from the few studies that have been done, they shed quite a bit of virus while they're sick, and that does tail off. When you're integrating it over a fairly large sewage area, I think it does seem to relate really well to cases. In smaller areas, let me think. In smaller areas, some folks have been doing a lot of really good research on university campuses where they have dorms and they can test the entire dorm and measure the wastewater coming out. And they've also found really tight correlations. If one person gets sick and they're measuring frequently enough, they can see that in the wastewater coming out of the dorm. So it seems to be correlated remarkably well, and it disappears reasonably quickly when it's that confined a group.
Thank you. That actually leads in well to a question here. Have you noticed faster changes, for example, in variants in certain communities like those without stricter precautions?
That is a great question, and I don't know the answer to that. That is something people are very, very interested in looking at. I know that they've noticed differences in the timing of when the variants come and when the spikes come even in the total virus numbers. But it seems like the variants it's more about the timing of when they're introduced than the precautions. The slope of the curve may be different from folks I've talked to. They think it rises a little bit faster and maybe plateaus a little bit longer. Those are the kinds of things I've seen.
Thank you. And then last question is any of your research published?
So some of our method comparisons are, we have a preprint out, and it should be, we're in revision in the Journal of Virological Methods. There's a few other of these papers are out, and I'm happy to forward those links to you all.
Please.
Yeah, happy to.
Okay, I think that's it for the questions that I see in the chat. If there are any others, feel free to add them. I am going to go back to Liz's presentation and add into the chat a link to the Bio-Rad video showing droplet generation. Oh,
Great, thank you.
I sent that to.
And thank you, Joshua. That was absolutely amazing. I can tell from the chat that people really enjoyed your presentation, and I have a feeling there are a lot more questions. But I look forward to reading about your research.
Well, I'm happy to have come and talked to you all, and I can speak for my colleagues. Like I said, this is only one of the things we use qPCR and digital PCR for, and I'm sure we'd love to come back and talk to you about other things too.
Great, thank you. Back to Bio-Rad. And I like that picture background you have.
Oh, thank you. That is Los Angeles Harbor. I think if I move my head, you can actually see a whale spouting.
Oh, really? Well, I just have to tell you really quickly, my mom lives on a cliff in Palos Verdes on Paseo del Mar. So we have a whole view of the ocean from my mom's house.
Yeah, this is on the San Pedro Palo Verde side, LA
Right.
Harbor. So yeah, right near Cabrillo Beach.
I grew up in San Pedro.
Really? Oh, that's awesome.
All right, thanks so much to both Liz and Josh. I hope everyone really enjoyed learning something about, I learned a huge amount about PCR that I don't know, and all the different applications. It's very interesting. I believe that Purnima was going to ask about surveys and things.
Yes.
I would like to just remind everyone that in 2 weeks, we'll have another presentation focusing on quantitative real-time PCR, and then a week after that, so Purnima will have the dates, I'm sure, and then a week after that, Linnea and Betsy Bodecker and Lee Brown will be talking about teaching advanced PCR techniques. So that's another 2 webinars in this series, and I would like to really thank InnovATEBIO, Cassandra, Purnima, and Linnea especially for helping us put on this series for everyone. All right, Purnima, over to you for surveys and things.
Thank you so much, Bryony. Thank you, Yolanda, and thank you, Joshua and Liz. This has been fascinating. We've had 2 of our 4-part series. Last week was our company showcase, and today has been a fantastic advanced PCR part 2 of our part 4. So we're looking forward to the next 2 as well. And as Bryony mentioned, our next one is going to be 2 weeks from now, October 29th. And the last one would be pedagogical and teaching methods of PCR. So we look forward to seeing all of you. And then as usual, please take the survey I'm going to post on the chat and help us bring you more such fascinating, wonderful webinars for the future.